Now I've established what optical isomerism is, I'm going to use this diagram to explain how we distinguish between them. So if we start over at this side here, we've got a cross-section of a light beam. So this is a light source, and light travels in all directions, as you can see from the arrows. And what they do is they pass this through a filter, which is known as a plane polarizer. So if you think of it with a slit in a certain direction, so in this case, imagine a vertical direction slit, and that's only going to allow this plane of light through. So the light that emerges on the other side is what we call plane polarized light, and these are only oscillating in this plane. Then if you imagine we shine that on our pair of optical isomers, so I've got a red one here and a blue one down here, just check you agree there's a chiral center there, and so we have optical isomer possible, and so we've got one form in the red box and the other form in the blue box. So remember, these are these non-superimposable mirror images of each other, and hopefully what you can appreciate is one of the isomers, so in this case the red one, is rotating the plane polarized light in a clockwise direction. The other isomer, the blue one in this case, is rotating the plane polarized light in an anti-clockwise direction. So a few years ago, some of my students went to Durham University to carry out a project on honey, which is a natural material, contains a lot of chiral molecules, and they were investigating the different chiral natures of different honeys. And so they made a sort of do-it-yourself polarizer. So if we have a look at this now, we've got a light source, which is just this torch here. And hopefully you can make out this sort of brown square above the lens there of the torch. That's the plane polarizer. Inside this boiling tube is the sample. So they were putting different honeys in there. The cardboard tubes, obviously to prevent any light from the lab from interfering with the experiment. And then they were standing looking down the tube through the protractor, and they were measuring the angle of rotation. And obviously different honeys gave different angles of rotation, and that enabled them to say how much or which chiral molecules were present in the honeys. So we'll just finish with this slide here. I've got an amino acid. So this is alanine with the R group of CH3. So you can see that we've got the clockwise rotation for the left-hand one, and we've got the anti-clockwise rotation for the right-hand one here. And just a bit of jargon for you, some terminology. The isomer that rotates plane polarized light in a clockwise direction is known as the dextrorotatory form. So you would sometimes see the symbol lowercase d, not uppercase d, lowercase d, next to the name of the chemical. So this is alanine. You would sometimes see D-alanine, or you may see a plus sign. The plus sign also symbolizes clockwise rotation. So either of those labels could be used. The opposite, the anti-clockwise rotation, is known as levorotatory, and so sometimes you might see a minus sign in front of the name of the chemical or a lowercase l. Now, just to confuse matters, there are uppercase D's and L's used. That's nothing to do with dextrorotatory and levorotatory.